In 1980, just over 30 years ago, Paul Berg received one of the most prestigious scientific research awards, the Nobel Prize in Chemistry, for creating the very first recombinant DNA molecules by combining the DNA of two different organisms. His recombinant DNA techniques opened the doors for other scientists to create recombinant DNA organisms by extracting these ring-shaped DNA strands from a bacteria's cytoplasm. These pieces of DNA, called plasmids, carry a few useful genes for the bacteria, such as antibiotics resistance. The Nobel Prize winners showed that the plasmid DNA can be extracted, reconfigured, and inserted back into other bacteria cells. This means that we can engineer bacteria cells to produce non-bacterial proteins that can be used in medicine, such as making human insulin, growth hormones, and hepatitis B vaccines, and in agriculture, such as vitamin A-enriched rice and insect-resistant crops. In the next few minutes, I will show you a simple lab procedure that extracts the plasmid DNA from E. coli bacteria. Here, we have 4 samples of E. coli in small Eppendorf tubes. The samples are placed in a centrifuge, an instrument that spins down the samples so the heaviest materials, in this case the bacteria, sink to the bottom. The liquid at the top is called the supernatant. Because we want our bacteria samples, we carefully remove the supernatant and we're ready to begin the isolation procedure. To extract the plasmid DNA, we will follow 6 simple steps. First, we'll suspend the bacteria, lyse the bacteria, precipitate out junks that we don't want, bind plasmid to the column, wash the column, and elute the plasmid DNA. In the first step, we want to resuspend the bacteria from the pellet at the bottom of the tube. To do this, we use a resuspension buffer. The buffer also contains RNase A solution, which degrades RNA that we wouldn't want in our solution because it is so similar to DNA. The second step is to lyse the cells, which means breaking down their cell membranes. We'll use a lysis buffer, which contains alkali that makes holes in the cell membranes, so that the plasma oozes out of the cell. The buffer also has a lyse blue reagent, which turns blue in the presence of DNA. If, after a couple seconds, the solution turns blue, then the plasmids in the cell cytoplasm have been released and everything is going well. Because we've just added alkali to the solution, the third step is to neutralize the alkali using a neutralization buffer. The neutralization buffer also precipitates unwanted proteins, lipids, and trapped chromosomal DNA, which form the white clouds in the solution and eliminate any sign of lyse blue. The tube is centrifuged for 1 minute at full speed, so all of the precipitate sinks to the bottom and the plasmids remain in the solution. Next, in the fourth step, we want to bind the plasmid DNA to a column. To do this, we add the supernatant to what's called a Chia-Prep spin column. The spin column contains a tiny silica membrane that binds to plasmid DNA. The extra liquid that does not contain plasmid DNA will pass through after centrifugation and can be discarded. The fifth step is to wash the column, to wash out non-plasmid DNA material stuck on the silica membrane. We add a wash buffer, which cleans the membrane and the plasmid sample. Finally, the last step to extracting the plasmid DNA is to elute or free the plasmid DNA from the membrane. The column is placed in a microcentrifuge tube that will collect the plasmid sample. We then add a elution buffer that detaches the DNA from the membrane. After we centrifuge the columns and tubes, the plasmid DNA has been spun down to the tube. Now we finally have our plasmid DNA, but we want to make sure that the product is pure. One way to check the plasmid DNA is by running an agarose gel. In the agarose gel, we see 4 clear bands that represent the plasmid DNA samples. This means that our DNA samples were pure and our extraction was successful. Now, the very first step of recombinant DNA technology is complete. Now that we have a purified sample of plasmids, we can insert new genetic code into the plasmids and introduce it to other bacteria, or study the genetic coding of these specific plasmids. From here on, the possibilities are endless. Thank you for watching this production from biolabscope.com.